Hello all, and welcome to our presentation titled Searching for the Northern Riffleshell Mussel. My name is Courtney Beals, and later in the presentation you will be hearing from my lab partner Sydney Thompson. A brief background of Sydney and I's involvement in Seneca Valley Center for Biotechnology Research. We perform eDNA studies with freshwater mussels. Last year we researched a mussel called the fat mucket, and this year we are looking at a mussel called the northern riffleshell, and we test water samples from western Pennsylvania creeks to see if these mussels reside in them. First of all, we need to establish what is eDNA. It stands for environmental DNA. Every organism leaves traces of their DNA in multiple forms, and eDNA can be extracted from the environment through water or soil samples, and this can be used to reveal the presence of a species without having to physically see it. And one of the great benefits of this process is that it is non-invasive, and this can be especially helpful when researching a federally endangered species because it will not disrupt their environment even further. Some background on the northern riffleshell. It is federally endangered, and it resides in French Creek, Conewago Creek, Allegheny Creek, and LeBoeuf Creek, and these are all local. So a big part of our process was creating a primer. And what is a primer? Well, it is a sequence of nucleotides that is the starting place for DNA synthesis. And to perform an eDNA study, it is critical to make a primer that is complementary to the nucleic sequences in the creature you are searching for, and in our case that was the northern riffleshell. And to do this, we target a mitochondrial gene. These primers are added with the experimental DNA in the PCR tube, along with a couple other substances such as master mix, which is the fluorescent containing Taq polymerase and nuclease-free water. And the qPCR will perform heat cycles to denature the DNA and to anneal the primers to the template DNA, and the Taq polymerase will extend this DNA out from the primers and synthesize new DNA. And this master mix chemical will only fluoresce when a double-stranded copy of the gene is created. The qPCRs can detect this fluorescence and display the results on an amplification graph. If there is no curve, or if the curve is dissimilar to the positive control, the creature's DNA is not present in your water sample. And this image depicts what the qPCR does and its heat cycles. Some gene qualifications we had to keep in mind. Well, the primer had to be unique to the northern riffleshell. The nucleic sequences in our primer targeted must not be similar to those in another organism, thus this would give us a false positive. And through multiple databases, we cross-referenced and found a gene that would do this job for us called cytochrome B. Once we found the primer, we could finally begin our research.
Hi, my name is Sydney. I'm Courtney's partner. I'm a junior as well. So I wanted to continue the presentation by showing the primer that we decided to use, and the gene in this primer is cytochrome B, which is a mitochondrial gene. So the steps to our experimentation were to filtrate the water samples we collected, homogenize the potential DNA found in those water samples, extract and purify the DNA, and then finally create a mega mix, which is probably the most important and instrumental portion of the steps before the qPCR, which in the megamix we had snippets of the primer and also a fluorescence containing Che master mix, and this would make the target genes, if found in the water samples, glow on the qPCR to show us that we indeed had a hit or a positive. So in trial 1, we actually did 2 sub-trials, and the only difference between these was lowering the amount of megamix we used, and this was just something that we learned we had to do for this experiment. And we did 2 sample locations and 1 control negative. So the first error we noticed was primer dimers, which primer dimers are potentially harmful because the polymerase could amplify the primer dimer instead of the actual desired gene, which could potentially create a false positive. And so we diluted the primers we used in trial 2 to hopefully fix this error. So here's a trial 2 graph. And I wanted to point out that although our trial 1 and 2 graphs look very similar, and this is because our technique and lack of contamination was very consistent, which is so good to roll out those potential sources of error. And so unfortunately, even though we tested more samples in trial 2, primer dimers did occur again. So we're hoping to continue our trials into the spring and eliminate primer dimers by just diluting those primers even further. And we hope to continue our studies with eDNA next school year with Chapman of the Western PA Conservancy, who can hopefully give us more samples than we have in our genetics lab library of water samples. And I just wanted to reiterate that freshwater mussels are important because they are filter feeders and they clean our water systems more than we realize. And they also provide shelter for many undeveloped organisms, just as they were once sheltered as parasitic organisms, as Courtney mentioned before. And eDNA is significant because it's a non-invasive way of discovering organisms and finding them, especially when organisms are endangered. Their habitats are left serene or as found. Even though certain populations, their demographics, such as how many young and how many geriatric portions of the organism's population are found, those can only be done through field work, but eDNA is still very important and innovative, and there's a lot left to be discovered. So thank you for watching.